Hello, everyone, and welcome to Chapter 26, which covers nervous system infections. As usual, we're going to start off by talking a little bit about the anatomy of the nervous system. The nervous system has two halves to it. There is the central nervous system, which consists of your brain and your spinal cord. And we can think about the central nervous system as being like the control center. Its responsibility is to receive and interpret sensory information and then send impulses back out to other parts of the body in order to react to that sensory information. Where that information comes from is the peripheral nervous system. So the peripheral nervous system is found in branches of neurons throughout the body. And these link the central nervous system to the body parts and allow the central nervous system to perceive signals in the environment. So it's probably no surprise to learn that the central nervous system is particularly important and that infections of the central nervous system tend to be more severe and life-threatening than infections of the peripheral nervous system. Let's take a closer look at the central nervous system, arguably the most essential body system, and therefore it requires special protections against microbial pathogens. So when we look at the central nervous system, brain, and spinal cord, they are covered in three layers of membranes that are called meninges. Those three layers are the dura mater, the arachnoid matter, and the pia mater. In between the arachnoid and the pia mater is the subarachnoid space that is filled with cerebrospinal fluid. The cerebrospinal fluid is tapped into your circulatory system, but it contains a very low concentration of elements of the immune system. And in general, the brain contains less presence of anything coming from the circulatory system because of something called the blood-brain barrier, which is the fact that the capillaries in the brain are less permeable than the capillaries in the rest of the body and let less stuff through. So this is important because it protects the brain. However, the permeability of those brain capillaries can change when there is inflammation, and this can sometimes allow things to get through that shouldn't get through, like pathogens. We have a little bit of terminology of nervous system diseases here. We have meningitis and encephalitis. Meningitis is inflammation of the meninges, those layers of membrane that protect the central nervous system. And encephalitis is inflammation of the brain tissue itself. So moving on to look at which diseases we're going to be covering, we have bacterial, viral, protozoan, fungal, and also something we haven't seen before, which are prion diseases. So we're going to start as usual with the bacterial infections. First of which is bacterial meningitis. So as we've learned, meningitis is inflammation of the meninges, so it's a central nervous infection. Bacterial meningitis, of course, is caused by bacteria, and of the two forms of meningitis, which are bacterial and viral, bacterial meningitis tends to be much more deadly and serious. It's associated with fever, headache, confusion, vomiting, neck stiffness, and then leading into the more severe symptoms and signs of coma, shock, and death. Sometimes, depending upon the pathogen and the immune status of the person infected, this can happen in just a few hours. It has a high mortality rate. Mortality rate is 15% if treated and 70% if untreated. And even if treated, 1 in 5 survivors usually live with lasting brain damage as a result of the inflammatory response that has occurred in their meninges. And as I said, bacterial meningitis progresses more rapidly and is more deadly than viral meningitis, although viral meningitis is more contagious. So bacterial meningitis can be caused by a wide variety of different species, but here we're going to look at the three most common. First of which is Haemophilus influenzae. This is a gram-negative coccobacillus-shaped bacterium. It was once the most common in the United States, but now it is much less common because of the HIV vaccine. the Haemophilus influenzae serotype B vaccine that is given to protect particularly children from this strain of bacteria. There is also Streptococcus pneumoniae. This species we've seen before. We've seen it as a cause of pneumonia and other infections. It is gram-positive diplococci in its morphology, and it is now the most common in the United States since the introduction of the Hib vaccine. And then lastly, there is Neisseria meningitidis. This is a gram-negative diplococci, and this one can be distinguished by a measles-like rash that arises on the skin and mucous membranes in conjunction with the meningitis. This leads us to our first checkpoint, where we are looking at a gram stain, which let's say was hypothetically prepared when a spinal tap was performed on a child with suspected meningitis, and the spinal fluid was smeared and gram stained, and the result is what you see here. So based upon the morphology and the gram staining reaction, what is the likely identity of the pathogen from the 3 common causes of meningitis that we just went over? Next, we have tetanus. Tetanus is caused by a bacterium called Clostridium tetani, which lives in the soil. It's an obligate anaerobe, and its gram stain reaction is gram-positive, and it's bacillus-shaped. This disease is non-communicable, meaning it is not contagious, and it cannot be transmitted from person to person. The portal of entry is deep skin wounds, usually with an item that has been contaminated with material from the soil that is carrying Clostridium tetani. So typically people associate tetanus with getting cuts from rusty nails. It's not the rust itself, it's not the nail, it's the depth of the wound created by the nail, and if it is contaminated with soil matter that contains Clostridium tetani, then if the wound is deep enough, then these obligate anaerobes will be able to access depth in the tissue where there is not oxygen present, and they can germinate and begin producing their main virulence factor, which is an exotoxin called tetanospasmin. Now, this can develop in a few ways. Localized tetanus is the less severe form, wherein the toxin causes contractions and spasms of local muscle tissue around the wound. Localized tetanus is what typically occurs among people who have been vaccinated as children, but failed to get their recommended boosters every 10 years. So they have some memory in their immune system, but it's not strong enough to completely ward off an infection. Generalized tetanus is what happens when the tetanospasm toxin enters the peripheral nervous system and then travels through the peripheral nerves to the central nervous system, resulting in violent systemic muscle spasms, and typically the muscles of the jaw are among those that are first affected, which is why lockjaw is associated with tetanus. And this can progress to affecting the cardiac and respiratory system, leading to death by asphyxiation. So the disease can actually shut down one's ability to breathe by affecting the nerves that are involved in respiration. Thankfully, immunization through the DTaP vaccine, which we've seen before, it's diphtheria, tetanus, and pertussis, where the T stands for tetanus, has made this disease extremely rare in the United States. However, like many other diseases, rejection of vaccine in recent years has arisen to some tetanus cases. For example, this one was a case where a boy had tetanus in Oregon who was 6 years old when he received a cut while he was playing on a farm and came down with tetanus. He had not been fully vaccinated against tetanus, and although his life was saved, the treatment was extremely intensive. It took months in the hospital and nearly $1 million in healthcare costs associated with saving his life. Part of what's so expensive about the treatment for tetanus is that it involves administering antibodies against the tetanus toxin that have been derived from human donor plasma. And so essentially, when you are in the hospital, you will receive an infusion of antibodies not created by your own body, but created by somebody else's body, and then those antibodies will help you fight the infection. So this leads us to a checkpoint question. What type of immunity is exemplified here? Is it artificial or natural? And is it active or passive? Next, we have botulism. Botulism is also caused by a soil microbe that is closely related to the microbe that causes tetanus. Clostridium botulinum is another member of the Clostridium genus, and it is an obligate anaerobe as well as a gram-positive bacillus, just like its cousin that causes tetanus. This guy produces a different toxin though. It produces the exotoxin called botulinum toxin, and the way that this toxin works is by blocking the transmission of nerve impulses, which leads to flaccid paralysis. It's associated with symptoms of drooping facial features, inability to open one's eyes fully, inability to speak and form words, and eventually the paralysis will affect again the respiratory and cardiac systems and lead to respiratory and cardiac failure and even death in severe cases. Botulism is very rare, but a handful of cases do occur each year, and they typically always involve food preparation methods that do not effectively kill Clostridium botulinum, which does naturally occur on food items because remember, it lives in the soil. And so what happens is endospores of these species are able to germinate in the anaerobic environment that is created in cans when oxygen is cut off, because that's the conditions under which it thrives. And so it's very important that industrial canning operations focus on killing Clostridium botulinum endospores. In fact, the entire industrial canning operation is based around how they can kill these spores. Their standards that have been in place are what is needed to kill these spores, and that includes high heat and pressure, acidity, and nitrates that prevent the growth of Clostridium botulinum. Nonetheless, infant immune systems are especially susceptible to this microbe, and this is why infants under the age of 1 year are not recommended to consume honey. Another interesting fact about botulism is that it is the foundation of the cosmetic procedure known as Botox. You may have heard of Botox as an injection that some people get in order to sort of stop the progress of wrinkle development on their face. And what is in that injection is a botulinum toxin, not the bacterium, but the toxin purified from the bacterium. And essentially, it creates microparalysis of nerves in the face, which then prevents the face from moving in ways that create wrinkles, because it is the natural movements of our face, for example, to smile and squint that lead to the progression of wrinkles. So by literally paralyzing the face to prevent this from happening, you can prevent wrinkles from developing. Okay, here's another checkpoint. The traditional Alaskan Native method of fermenting fish heads is to place them in a grass-lined hole in the ground, like what you see here. The CDC recommends that to prevent botulism, the traditional method continue to be used rather than burying the fish heads in sealed plastic containers, which is what some people have done. But it's not a good idea. So in what way does the traditional method prevent botulism as compared to placing fish heads in sealed plastic containers? This brings us to our final bacterial infection, which is leprosy, also known as Hansen's disease. It's caused by the acid-fast bacterium Mycobacterium leprae. So this is a species that has that waxy cell wall which offers additional layers of protection. They colonize the myelin sheaths that surround peripheral nerves, and they grow best at cooler temperatures. So not your core body temperature, but the cooler temperatures found in your nose and your toes and your fingers, which is somewhat lower than your core body temperature. It is not a very contagious disease despite what may have been passed down from ancient times. The idea that leprosy requires quarantining to prevent its spread is not accurate. It's only transmitted through prolonged and repeated direct contact. The manifestation of the disease depends upon the strength of the immune response of the person who is affected by it. People who have strong immune systems will come down with tuberculoid or neutral leprosy. These are two different designations used by different organizations, but they essentially mean the same thing. And this is the milder form of leprosy that results in discolored skin regions that are associated with a loss of sensation, such as what you can see here in the upper photo. The more severe form of leprosy occurs in individuals who have weakened immune systems, and that's lepromatous leprosy, also known as progressive leprosy. And this is what you probably associate with leprosy in general, although it's not as common as tuberculoid leprosy. The result of lepromatous leprosy is disfiguring nodules across the body, especially in the nose and the hands, where the bacterium tends to colonize because of its preference for lower body temperatures. So we've finished running through our diseases that are bacterial in origin, and we're going to move on to talking about the viral infections, starting with polio or poliomyelitis as it's called. This is caused by the poliovirus, which is transmitted through water that is contaminated with sewage. So technically, it's the fecal-oral route. Most cases of polio are asymptomatic or cause mild intestinal distress. However, in a small percentage of cases, the virus disseminates from the tonsils to the lymph nodes into the bloodstream, where it makes its way to the central nervous system. And in these cases, the virus will replicate in the motor nerve cells, which can kill those cells and lead to loss of function, paralysis, and death. So polio used to be a disease that was responsible for paralyzing many people, including large amounts of children around the world. And today, owing to a very aggressive vaccination campaign since the development of the vaccine in 1955, polio is nearly eradicated worldwide. It is very, very close to being the second human disease to be eradicated after smallpox, which we learned about in our skin infections chapter. The only places where polio is still present around the world are Afghanistan, Pakistan, and Nigeria. And the reason why that's the case is because the World Health Organization's Global Polio Eradication Initiative has been disrupted there by war and political instability. But this is still a work in progress, and hopefully not too long from now, we will see polio eradicated. And I wanted to show everyone this figure as well, which is a graph showing the rates of paralytic polio cases in the United States over the years. So this is not total polio cases. These are the numbers of cases of paralytic polio, so polio that progressed to paralysis. And as you can see, a peak was reached in the 1950s, where there were well over 50,000 cases annually of paralytic polio. In 1955, this is when the polio vaccine was developed, and the rate of paralytic polio and polio deaths dropped substantially after that point and has remained extremely low ever since then. So we can see what a huge impact the polio vaccine has made since 1955. Our second viral infection is rabies. Rabies is caused by the rabies virus. You can see an electron micrograph image of the rabies virus in this upper photo. And it has a unique bullet-like shape associated with it. You probably already know that rabies is transmitted through animal bites, and the most common reservoirs in the United States include raccoons, bats, and skunks. The domestic animal reservoir is pretty non-existent now in the United States because of vaccination. Wild animals, interestingly, are also vaccinated through projects that airdrop feed that has been laced with an oral vaccination leading wild animals to consume that vaccination and become protected against rabies as well. So this also contributes to the fight against rabies. The rabies virus has an extremely long incubation period, and that's because it travels painstakingly through the peripheral nerves at a rate of 15 to 100 millimeters per day after exposure. And so this means that after a person is bitten by an animal, there is a period where they do not experience symptoms before it reaches the central nervous system. And this means that post-exposure vaccination and antibody administration from a donor is effective as a treatment, because this delay before the virus reaches the central nervous system allows your body to react to the vaccine and develop antibodies against it. And in that way, once it reaches the central nervous system, your immune system can take over and eradicate the virus. So yes, there is a rabies vaccine available. It's not given unless you work closely with animals for a living, or in the cases of having been bitten and needing post-exposure vaccine to protect you before the virus reaches the central nervous system. However, once the virus reaches the central nervous system and symptoms begin to appear, then the disease has a virtual 100% mortality rate. At that point, nothing can be done. There is no effective, proven, demonstrated treatment for rabies. There is something called the Milwaukee Protocol, though, that involves putting a person in a chemically induced coma for many months and administering antiviral drugs. However, this protocol has only been demonstrated to be effective a handful of times, and there are only a handful of people who have ever survived rabies infections without getting post-exposure vaccination. Next is arboviral encephalitis. So this is not a single disease, it's a group of infections that are caused by arboviruses. And arboviruses is a word that is used to refer to arthropod-borne viruses. So it's a mashup of these three terms. Specifically, it refers to viruses that are transmitted through mosquito bites. And so in the United States, there are 4 arboviruses that are endemic here. And those include Western and Eastern equine encephalitis, St. Louis encephalitis, and West Nile virus. The most common one is West Nile virus. West Nile virus is the leading mosquito-borne illness in the United States. And so I've included an image here that indicates the number of cases per 100,000 people, where darker means higher number of cases. So you can see the range of the West Nile virus infection. These infections can range widely from asymptomatic to mild, which includes chills, headache, and fever, to severe, which is associated with coma, neurological damage, and death. The majority of people who experience severe infections are people who have compromised immune systems or are older and therefore have experienced immune system decline. There are no antiviral drugs available to treat these infections. Treatment when these infections occur is merely supportive care, and so the best way to prevent them is through mosquito control. Our last viral infection is the Zika virus. So this is an emerging arbovirus, meaning that it is an arbovirus that has increased in prevalence and it's ranged over the last few years. It's endemic to tropical regions, but that range is expanding along with the range of the mosquitoes that carry it. Most cases of Zika virus are mild, including a fever, a slight rash, and perhaps some conjunctivitis. However, infection of a woman while she is pregnant can lead to serious brain damage and birth defect called microcephaly in the developing fetus, which is where the head does not develop to the normal size, which of course impacts brain size as well as comes with intellectual disability. There is no treatment and there is no vaccine for Zika virus at this time, although those are both still under development. So at this point, we have another checkpoint. A patient presents with a fever, headache, and behavioral agitation and confusion. 2 months earlier, the patient had been bitten by a feral cat but did not seek medical attention. What is the likely causal agent and what is the treatment? And I'll give you a hint. This is a little bit of a trick question. So that completes our look at viral infections. We have one fungal infection that we need to take a look at, and that is cryptococcosis. Cryptococcosis, this is the only fungal form of meningitis. It's caused by the species Cryptococcus neoformans, which is a yeast-like fungus that has a unique virulence factor, which is its capability to form a capsule. This capsule has a protective effect for the fungus against our immune system, which is what it owes its ability to initiate meningitis to. Without this capsule, it would not be able to cause meningitis. Cryptococcosis is a rare disease. It can manifest as a respiratory infection, which is transmitted through inhalation of dried bird droppings, typically those of pigeons, but it can also progress from a respiratory infection to meningitis by traveling through the bloodstream. And although healthy individuals usually experience a subclinical infection, this infection is particularly dangerous for immunocompromised individuals, especially those who have the third stage of an HIV infection, AIDS. And so oftentimes, if a person with AIDS experiences a cryptococcosis infection, it requires antifungal treatments that goes on for months, and recurrences of this infection are frequent. Next, we have 2 protozoan infections to cover. The first of these is African sleeping sickness, or African trypanosomiasis. This is caused by the protozoan, which you can see in this lower image here, called Trypanosoma brucei. It is transmitted through the bites of tsetse flies, the fly that you can see in the upper image, and it is a parasite that enters through the blood. The initial signs include a fever that comes and goes, and as the disease progresses, it includes symptoms such as headaches, seizures, slurred speech, and increased amounts of daytime sleeping, which is why it is referred to as African sleeping sickness. The pathogen has the virulence factor of antigenic variation, which makes it a difficult infection to get rid of. Even as the immune system is clearing it from the body, subsets of the pathogen will vary their antigens and evade the immune system while the infection is still ongoing. The current approach to preventing it is vector elimination rather than vaccination, that is, getting rid of the tsetse flies. Next, we have amoebic meningitis, which is colloquially referred to as the brain-eating amoebas. There are 2 varieties of amoebic meningitis. The first is just referred to as standard amoebic meningitis, caused by the species Naegleria fowleri. The portal of entry for this pathogen is through the nasal passages. It resides in warm freshwater sources, and people typically acquire this infection while they are swimming and get water in their nose. From the nasal passages, the amoeba moves to the olfactory nerves, and then it travels through them to reach the subarachnoid space between the layers of matter in the meninges, and within a few days, symptoms emerge including confusion, seizures, coma, and eventually death. It has a near 100% mortality rate within days, and the pathogenesis moves so quickly that oftentimes diagnosis does not take place until after death, in most cases through autopsy. To give you an idea of how deadly this is, only 3 out of the 138 patients who have experienced this disease in the US have survived. There is another special type of amoebic meningitis called granulomatous amoebic meningitis, and this is caused by members of the genera Acanthamoeba or Balamuthia. So these guys also enter through the nasal passages or through skin wounds, and this develops more slowly. It's a subacute development over weeks or months, but it still has a high mortality rate. So unlike standard amoebic meningitis, which develops over days to a near 100% mortality rate, granulomatous amoebic meningitis develops over weeks or months, but has an equally high mortality rate. And in this checkpoint, now we've heard about acanthamoeba before. Tell me what other disease we've learned about is caused by acanthamoeba. We're almost to the end here. We have the prion diseases left to cover. Spongiform encephalopathies, as we've discussed briefly before when we covered acellular pathogens. But let's review how prion diseases work. So neurons in the brain naturally express this prion protein, which is designated prion PRPC on their surface. This is a normal part of human brain cells. However, people can develop mutated forms of this prion protein, and the interaction between the mutated, misfolded prion protein is able to corrupt normal prion proteins and cause them to also be misfolded. So this reaction propagates, and as the mutated proteins corrupt the normal prion proteins, they develop into plaques in the brain. And these plaques cause the brain to become porous like a sponge, leading to severe degeneration. Prions are resistant to all different types of disinfection. They do not respond to boiling, irradiation, or autoclaving. So they are a really scary little acellular pathogen. And we're going to talk about two different types of spongiform encephalopathies, one that infects humans and one that does not. The first of these is bovine spongiform encephalopathy, which is colloquially called mad cow disease or scrappy. It affects deer, sheep, and cows, and the acquisition to domestic cattle is spread through consumption of cattle feed that contains this protein in it. So they eat the misfolded prion protein and then they acquire the disease. So where does the misfolded prion protein come from? How does it get in their feed? Well, the feed can be contaminated with prion protein because other animals are processed and fed to the cattle. So yes, it's kind of hard to wrap your head around it, but herbivorous cattle are fed feed that contains bone meal and other types of meal that comes from processed animals. So when it's contaminated with a prion protein, that's how it can spread. The mad cow disease outbreaks that have happened historically have always been controlled by culling the affected animals and stopping the infection by simply eliminating animals that have this infection. In humans, there is an infection with a similar pathology called Creutzfeldt-Jakob disease. This affects humans at a very low rate. There's approximately 300 cases that occur in the US every year, so it's a rare disease. It has essentially the same pathogenesis of bovine spongiform encephalopathy in that it involves the development of plaques in the brain that lead to a spongy texture. And the degeneration is associated with dementia, insomnia, dysphagia, which is the difficulty of swallowing or speaking, and motor control problems. It is known that this disease runs in families and that there are genetic risk factors for it, but it has also been transmitted, and it has been documented as being transmitted through corneal transplants from a person who is affected to a person who is not, as well as through contacts with the wounds of people who have this disease. So even if you don't have the genetic risk factors for it, you can acquire it from someone who does. It is fatal within a year of symptom onset, so it's a very serious disease, and again, there is no way to treat it. There are no drugs that treat prion infections, although there is research in this area. And this brings us to the end of our final lecture on infections of the nervous system.